In this video, I will explain you about the digestion process of the protein. And let's assume that we eat hamburger again, and inside the hamburger there are more or less the proteins as well as fat and carbohydrate. And in this time, we'll discuss about the digestion process of the protein. So protein will be in our mouth. And unfortunately, the saliva has no proteolytic enzymes, so protein just moves into the stomach without digestion. And inside the stomach, protein is digested into large polypeptide, and this process is done by pepsin. And pepsin is secreted by the chief cell in the gastrium, and chief cells secrete the zymogen, the inactive form of the enzyme called pepsinogen. And this pepsinogen is converted to pepsin, and this pepsin performs the digestion of protein into large polypeptide. And pepsinogen is converted to pepsin, and this conversion is done by hydrochloric acid secreted by parietal cell in the stomach. So the hydrochloric acid activates the pepsinogen into pepsin, and also it provides the optimum pH for the pepsin to work properly. And this large size polypeptide moves to the small intestine by the process of gastric emptying. And to maintain consistency, we represented it like this. And this gastric emptying also moves the other substances, digested substances. For example, this gastric emptying moves the polysaccharide in the stomach or oligosaccharide in the stomach into small intestine, and also this gastric emptying moves the triacylglycerol in the stomach into the small intestine. And this large size polypeptide will be digested into small size polypeptide. This is done by trypsin and chymotrypsin, the enzyme secreted by the pancreas. But the pancreas does not secrete these enzymes as an active form again. Pancreas always secrete the enzyme in a zymogen to prevent its own digestion. And we have encountered the trypsin before in the lipid digestion part. The trypsin here is activated from trypsinogen by enteropeptidase, the one of the brush border enzyme in the surface of the enterocyte. And this trypsin was acting on the procholipase, which are converted into cholipase, and as the helps the lipid digestion. And trypsin also helps its own conversion, its own activation. And here trypsin also have a role as the activation from chymotrypsinogen into chymotrypsin and procarboxylase into carboxylase. So trypsin has a total 4 roles until now. It activates its own activation and activation of procholipase, which is involved in the lipid digestion, and activation of procarboxylase and chymotrypsinogen. So the trypsin has many important roles in our lipid and uh, protein digestion. With the help of trypsin and chymotrypsin, large size polypeptide is digested into small size polypeptide. And this small size polypeptide will be finally digested into dipeptide and into amino acids. And this digestion is performed by aminopeptidase, carboxylase, and dipeptidase. Among them, aminopeptidase and dipeptidase are the one of the brush border enzyme on the surface of the enterocyte, and carboxylase is activated form of procarboxylase. Its activation is performed also by trypsin. And finally, amino acids in the small intestine will absorbed by the enterocyte. I will go into more detail about that later. Thank you for listening.